Slugs and snails are seemingly harmless critters, but they play an important role in many disease transmission systems. One example is brainworm infections in moose, which is the topic of my infographic. Minnesota moose populations declined by around 60% over the last several decades. Studies to determine the primary causes of the decline were recently completed by the Grand Portage Department of Biology and the Minnesota Department of Natural Resources. They independently concluded that brainworm infection was the top cause of death in Minnesota moose, accounting for about 24 to 33% of mortalities. Moose are important to Minnesotans from a cultural, economic, and ecosystem health perspective. Culturally, moose hunting is a treaty right and an important source of cultural preservation for Minnesota Anishinaabe groups, such as the Grand Portage Band of Chippewa, our project partners. A quick perusal of any Minnesota-themed gift shop should also convince you that moose are central to Minnesota's cultural identity, since moose images are on most items in those stores. Economically, tourists are drawn to rural Minnesota communities by the possibility of seeing moose, boosting these economies. From an ecosystem health perspective, moose grazing structures aquatic environments with cascading effects for other species that share that environment. So how does a snail or slug infect a moose with brainworm? The complex cycle starts with an infected white-tailed deer shedding brainworm larvae into the environment through feces. Importantly, white-tailed deer are the natural hosts of brainworm and unaffected by infection. Slugs and snails are attracted to the feces by the moist microclimate and become infected through their foot's mucosal membrane. The infected slugs or snails are then ingested by a deer, in which case the cycle starts over, or by a moose, whereby the brainworm becomes lost in the moose's body, often leading to neurological symptoms and, unfortunately, moose death. Although much is understood about the deer's role in transmission, little is known about the slug or snail hosts, including which species are important to the cycle. A logical next step, then, is identifying which slug and snail species are important for transmission. We can explore this question by observing which slugs or snails moose eat. To do this, we use a method called DNA barcoding, which works similarly to checking out at the grocery store. When you pick out a grocery item, you'll notice that it has a barcode tag. When scanned, this barcode links to a database containing that barcode and the corresponding price for the item. Similarly, certain portions of the genome are unique identifiers for each biological species. Specifically, a portion of the genome called COX-1 is an excellent quote-unquote barcode for species identity. Because of this fact, databases have been compiled that contain species and corresponding COX-1 sequences, which serve a similar purpose to the barcode and price databases in grocery store computers. When you extract, amplify, and sequence COX-1 DNA from a sample, you can match the COX-1 sequences from the sample with COX-1 sequences in the database to get an idea of which species are in your sample. When we do this with moose feces, we find out which species of slugs or snails were recently consumed. The findings of this effort will have several implications for moose. First, by knowing which slugs and snails are important and understanding their ecology, we can modify the landscape to reduce the abundance of those species in moose range. Similarly, it is unclear whether many species of slugs or snails are even capable of carrying brainworm. Knowing which species moose are eating will allow us to focus future studies on carrier competency in slugs and snails. Finally, with a more complete knowledge of which species of slugs and snails are important for brainworm transmission, we can better predict infection risks for moose. Thank you for taking the time to learn about this important topic. If you'd like to help us save Minnesota moose, please contribute to our research by scanning the QR code, which, by the way, is another example of barcoding, with your phone's camera or QR scanner and making a donation. We truly appreciate your interest and support. Thank you.